Hello everyone, welcome back to another BIO2060 lab meeting. So today I'll be going over your assignments that are going to be due in 1 week on 4/20. They're going to be due at 11:59 p.m. and there's going to be a couple things you're going to have to do. So you're going to have to be completing your interactive assay chart assignment, which is using this interactive website here. So this here is a Kirby-Bauer antimicrobial susceptibility test, and you're going to be testing the effect of each antimicrobial antibiotic on here. So we have amoxicillin as the first one, which is going to be an AMC on the disc. So here where we see AMC, we're going to be testing how susceptible this organism is. So how this test is normally performed: you take your plate and then you streak your organism completely on there. You fill up the plate with growth, and then you place these discs that contain the antibiotic or antimicrobial agent that you're testing, and then you're going to have these rings of inhibition, these zones of inhibition where there's no growth, because this antimicrobial agent is stopping the growth of this organism. So when you incubate it, there will not be any growth around the things that the organism is susceptible to, and then the ones that they're resistant, they're going to be able to grow right next to it, and they're not going to be affected by this antimicrobial agent at all, or very little. So with amoxicillin, what we'll do is we'll take this, and then we'll see how many millimeters it is. So when we are measuring this, we can even zoom in. So when we measure this amoxicillin, it gives us about 12 millimeters, right? So it's a little bit more than 1 centimeter. We're about 12 millimeters here. So you do the diameter. So from one side of the circle of inhibition to the other. So we go from here all the way to here, and we get 12 millimeters or 1.2 centimeters. So in that case, when we check amoxicillin, that's going to be less than or equal to 13, so 12 is going to be less than that, so we'd select resistant. So then you just go through and do that for all of these. So then if we check cephalothin, which is going to be a CF, we're going to go, so we find the CF, we check it, and then we get our ruler, we place it at one end of the zone of inhibition, and then we are going to measure that, and that gives us about 18 milliliters. Or millimeters, okay? So then that's going to be 18, greater than or equal to 18, that is susceptible, okay? So the bigger this zone, the more susceptible the organism is to this antimicrobial agent, okay? So once you've filled up all of these, what you're going to do is you're going to go here and you're going to take this assignment, little quiz assignment test. So once you do it, all you need to do is just put in whether or not it's resistant, intermediate, susceptible, and you'll have 5 minutes to do all that. It should be very easy. You'll have all the answers saved right here, so you can just go back and forth and put them all in, and then submit it. And if you do, well, hopefully you'll do better than I did, because I didn't get any of them. That's awkward. So each one is going to be worth half a point, and there's going to be a total of 6 points for that assignment. So if we go back, you have one other assignment, which is going to be Quiz 5, which is going to be a combination of multiple choice and true-false questions based off of this coronavirus video right here.
In December 2019.
And then also going to be based off of the Lab 10 lecture, which is going to take place right now. So, Lab 10, from the last lab, we're basically just going to be observing the effects of temperature, pH, salt, oxygen on the different, on our unknowns in different organisms to figure out whether they grew or didn't grow. So for Lab 10, what we're going to be doing is testing how we control different microorganisms. So we're going to be, last time we checked how the different optimal growth environments change by affecting the temperature, osmotic pressure, pH, and oxygen content of the atmosphere. So today we're going to be testing the chemical methods and the physical methods, or we would be testing technically. So the different levels of chemical controls, we have sterilization, then disinfection, and antiseptic. So these are differentiated because sterilization is going to be the elimination of all of the viable microbes. That's going to be including spores. So sterilization is going to be the elimination of everything, all the different microbes, spores, everything that's going to be on there. And then disinfection or decontamination is going to only eliminate the pathogens, or so it's not going to be all of the viable microbes, and this is going to be from a non-living object or surface, okay? And it's not going to completely eliminate all microbes. It's just going to be nearly all of them. So that's like the 99.9%. Then we have antiseptic. So this is an agent that kills or inhibits microbial growth, but is not going to be harmful towards human tissues, okay? So this can be applied to living surfaces, right? So this is a big difference between disinfection or decontamination and antiseptic. So an antiseptic is going to be used for living tissues. You can put it on your skin. Okay. So germicides are disinfectants. They're going to act by targeting at the microbial cell envelopes, and then they're going to be denaturing proteins. So germicide is what we use quite a bit in this lab. And then antibiotics. So these are going to be natural or synthetic therapeutic drugs, and they're going to be used to treat bacterial infections. So they can be used in humans, animals, and plants to help treat them. And they're going to interfere with different cellular processes. So whether that's creation of the cell wall or protein synthesis or DNA replication, a bunch of different things they can interfere with. So first thing we'd be testing is how effective are these germicides. And, well, we're not doing that, unfortunately. We all know why. So how effective are antibiotics in controlling microbial growth? So antibiotics are just naturally or chemically synthesized compounds that can stop the growth of or kill bacteria, right? So they can stop the growth of bacteria or they can kill the bacteria. Those are two different ways, right? If we stop the growth, it's going to have the same effect. So then we have bactericidal, which is an antibiotic that kills bacterial cells. And then we have bacteriostatic, which is going to prevent the growth of bacterial cells, okay? So static is in non-changing, and then cidal is in to kill. So other microbes are going to have different drugs. So with viruses, we have antivirals. With fungi, we have antifungals. So antibiotics are for bacteria. Okay, so the thing that we're going to be using to test the effectiveness of the antibiotics is the Kirby-Bauer test. And so that's going to be your interactive assay that you're going to be testing. You have that assignment that I gave you, so that's going to be kind of making up for this assignment right here that we can't do in the actual lab, unfortunately. So UV radiation is also going to be another physical method that we can use to control microorganism growth. And so this is going to work by targeting the DNA, and it's going to form thymine dimers. Okay, so basically it's just going to combine them together like that. So instead of having two separate thymine, they're going to be joined together, which is going to cause some major issues for the DNA. And then it's also it's going to be able to penetrate, not very much, so it's going to be only on like a surface layer penetration. And then this isn't going to be allowing for sterilization. So this will be for decontamination, but not so much sterilization. You're not going to be able to kill everything with this. And then we also have filtration. So you're going to be using a filter to try and stop the organisms from getting through. So the pore sizes are going to be pretty small. So it's going to stop any bacteria or larger from getting through the filter, and they're going to be stuck on the filter. So you'll be able to filter them out that way. However, this won't be able to work with smaller organisms like viruses, unfortunately. So this is using UV radiation here to help decontaminate, and this is what we use in our vent hoods. So the most effective method of killing microbes is going to be heat, okay? So not all methods of heating are going to achieve sterilization, but there's at least one method that we have, which is going to be autoclaving, right? So we've talked about this before. So at 121 degrees Celsius moist heat and 15 psi is going to allow us to achieve sterilization. Then we also have fire incineration, which is going to kill most organisms, but not sterilization, right? Not going to kill all of them. Then we have dry heat, boiling water, and then pasteurization. So pasteurization is going to be using a lower temperature to kill the majority of pathogens so that it's safe to eat, but it's not going to completely sterilize the food. Okay, so this is a picture of autoclave. Looks kind of old, but they look a little bit newer now. Then the next thing we'd be doing is testing how UV radiation affects microbial growth and whether it can completely eliminate it. So unfortunately we're not doing that, but hopefully you kind of got an idea of physical methods we can use to stop microbial growth. So you don't need to record this, of course, because we're not doing it. So sad. So with that, that's pretty much everything. Just make sure you do all the assignments that I posted. They'll be due 4/20 at 11:59 p.m. So make sure you do everything, and with that, we're all done. See you next week.